And no further comment. Yes, my lip looks jacked up. That's what I get. Hey, you know what they say about Fight Club? Don't talk about Fight Club. Look at these eggs. Ooh, look at this first dozen. Well, these are the dozen that I haven't eaten yet. Look at these green ones. We got a new green one today. It's way smaller. And you know how I know it's new? Da-na-na. Y'all see that? You know what that is? That's blood. You know where there's blood there? Because this egg came out a cloaca that wasn't very big. Yeah, maybe it is now. Anyway. I mean, just think about if we laid eggs that were this proportional to our bodies. Just think about ostrich eggs. Oh my goodness. Because ostriches are like kind of like our body size. Just think about how big their eggs are if we had to lay eggs. Hold on, I may have an ostrich egg here. Yeah. So they're like, whoa, what's a little egg coming out? I go to the bathroom all the time, something like that. Well, you know what you don't normally come out, or doesn't come out, something this big. It's an ostrich egg. Can you imagine that, Joker? Well, I don't have to lay eggs. I think it's ginormous. Now, this probably isn't, that's probably about the size of a baby's skull, right? So someday when you ladies have kids, then, you know, this thing will, you get the idea. Backstory. So I went to South Africa one time hunting, and I found this ostrich egg out in the bush, and it was rotten. Let me tell you, this joker was rotten and nasty and stinking. And I paid our tracker. I was like, how much you charge me to clean that thing out? Because I knew he had ways. They sell these things. And so I cracked a little hole in the end, sucked all of that rotten egg juice out of this gigantic egg for $5. And I was like, I'll take that one. Can't believe I got this back home without it cracking though. Anyway, it's beautiful. It's beautiful. All right, today let's actually learn something. We are on part 5 of Mendelian genetics. You guys need to get out your pedigree from yesterday where you were tracking hemophilia in the royal family. I gave you a list of possible genotypes that you could use, and you were supposed to fill those in for all of the individuals on the pedigree. Now, if our friends at home will come with us here, don't worry about what that drawing is. It won't make sense. These are the possible genotypes, with the H's being the hemophilia on the X chromosome. Now, on our pedigree, and those people of you at home are trying to work through one of these, just look up pedigree, hemophilia in the royal family. This should come up. The easiest individuals on here to label are what? The males. The males with it shaded in, because we know they're what? They have hemophilia. So if they are shaded in, we know they have hemophilia for sure. But also the other males are super easy because we know they are males that are not carriers. And so that's pretty easy to figure out. We've only got 2 choices for males up here. So those are super easy. And if we had any females that were shaded in, they would be really easy to identify too because they would have hemophilia genes. The hard ones are to decipher are the females that don't have hemophilia, and so that's all females here since we don't have any that have hemophilia. And you guys have to decipher, are they carriers or not? Or could it be either or? Maybe you can't narrow it down. So that's the difficulty in this family tree. So remember, the pedigree. When you're making a pedigree, always do the easy stuff that you know first. The ones that you can identify, go ahead and throw those genotypes up, and then you can use those to help figure out the other things. Okay? So, we'll go over that later, maybe even Monday if we don't get to it, because today we have a lab. It's the genetics lab, historic, I think lab 6. And we will be starting on that. And there's a lot of genetic analysis that we'll be doing in that lab. I really like it. You're supposed to breed fruit flies for it, which is a total pain in the butt. So I've not done that since the first year because I made that mistake and I had fruit flies running around here for like 7 years. And that's no joke. I mean, I was killing fruit flies forever. And so I'm going to give you some sample data and we're going to analyze that because that's probably what you would have to do on the AP exam anyway. But before we get there, let's turn to page 298. Now, yesterday we talked a little bit about chromosomes, And X and Y, we talked about sex-linked genes, which is fun to discuss since we all have 23 pairs of chromosomes, and that 23rd one is either XX if you're female or XY if you are male. Now, ladies, both of those X's aren't always turned on. And in fact, most of the time, one of them is turned off. Did y'all know this? So I want you to add in at the end of part 4 notes this thing called bar bodies. B-A-R-R bodies. Dun-dun-dun. Now, of course, this is named after some guy named Barr. So we've got some cats over here, and then I've got a little paragraph that we're going to read through. So if you guys want to listen along up here in the second paragraph. And let's talk about what Barr bodies are real quick. Females, mammals, including humans, inherit 2 X's, twice the number of males. In fact, almost all of one chromosome in each cell in a female becomes inactivated during the embryonic development. So normally one of those is switched off. As a result, the cells of females and males have the same affected dose of most X-linked genes. An inactive X in each cell of a female condenses into a compact object called a Barr body, named after Barr. Most of the genes on the X chromosome that forms the Barr body are not expressed. In the ovaries, however, the Barr body chromosomes are reactivated to cells that give rise to eggs. So that's kind of the exception where you have both of those X chromosomes. Now, why does all this matter? Who cares? Does it have anything to do with anything? Well, in fact, for those of you cat lovers out there, if you have a calico cat or if you have a tortoiseshell cat, I'm going to be honest, I've never heard of a cat like this named tortoiseshell. Have y'all? That color? Matthew's the only one in here. So this color right here, or you can also apply this to calico, but there's one caveat with calico cats. Those cats are always going to be, yes, but they're always going to be, which sex? Female. Inactivation, X inactivation in the tortoiseshell cat. The tortoiseshell gene is on the X chromosome, and the tortoiseshell phenotype requires the presence of 2 different alleles, one for orange fur and one for black fur. Normally, only females can only have both those alleles because they have both X's. And if a female cat is heterozygous for the tortoiseshell gene, meaning she has one X that codes for orange and one that codes for black, orange patches are formed by populations of cells in which the X chromosome with the orange is active. Black patches have formed where cells have the X chromosome where the black allele is active. Calico cats also have a white areas which are determined by another gene. But the point being, basically when you see these multicolored cats like that, the tortoiseshell and the calico, where you have this orange and black gene that's located on the X chromosome, the reason for that is you have one gene you got from mom, one gene you got from dad, one is for black fur, one is for orange fur, and some of those X's get turned on in one location that codes for orange fur, and then some X's get turned on in another location that codes for black fur, and you wind up with this multicolored looking cat, okay? You're not going to see that in boys because
boys only have one X.
you only got one X, so you're usually going to be the orange or black cat. So, take note of that, ladies. All right.
So what can we
Cats, anyway.
Just like summary of Barr bodies.
Exactly, where one X chromosome is inactivated is the main thing there. All right, let's skip on to chromosomal errors. Now, we've talked a little bit about chromosomal errors in the past. I know y'all probably heard of Down syndrome. It's the most prevalent to show up where humans are fairly normal and functioning, although there's some phenotypic problems that result in Down syndrome. There are some other chromosomal abnormalities we're going to talk and work our way through. Some of y'all may know someone with one of these, but these provide some problems because of this extra chromosomal input. Now, normally what happens here, if you look at page 305 in your Campbell book or whatever you're looking at, I'll show you guys at home. The problem that results where you wind up with an extra chromosome or you're missing a chromosome is this function called non-disjunction, meaning the chromosomes don't separate. And it's either not going to separate at which 2 points. There's 1 or 2 places where this could occur.
Meiosis 1 and meiosis 2.
But specifically when during meiosis 1 or meiosis 2? Anaphase. So either during anaphase, which is the one on the, anaphase 1 would be the one on the left side. And anaphase 2, well, I think it's backwards for you people looking here. That's where anaphase 1, the homologous chromosomes don't separate. And the other one on the right side for us here at school, or I think it'll be the left side for you guys, is where the sister chromatids don't separate in anaphase 2. And because of that, you wind up with chromosomal abnormalities in the resulting cells. Some have an extra one, some have one less, and it depends on, if you're making sperm or egg and passing those on, then that's going to determine whether you have an extra chromosome or one less. Now, there are some examples of this. Down syndrome is one. We call it trisomy 21 because you have 3 21st chromosomes, where you should normally have 2. Why is trisomy, do we have other trisomies like trisomy 10 and 15 and 1 and 3? You ever hear of those? Why not? Do they occur?
Because
Yes, they do. Why do you never hear of them?
less likely.
I don't know if it's less likely. That's a good question. Here's what I know. 21st chromosome is the smallest one. Contains the fewest genes. And it's going to have the least impact having that extra DNA. And so the other ones, especially when you get toward like 1, there's a lot of DNA. I mean, these are really big chromosomes. And so there's more stuff that gets affected. Most of the time, that would result in a child, what? Yeah, I mean, it probably wouldn't develop and result in a miscarriage or death early on because of that. You can look up on like Wikipedia or wherever, Google it, like any trisomy, like trisomy 1, 2, 3, 4, whatever. And I'm sure they probably have names given to them. As you go down the line, some I think are more common than others. And some you can actually see kids that have developed to a certain point, and some of them are very disfigured and developed in not a normal way. And so that's why you normally don't see those other types of chromosomal disorders in the population. Now, there are some sex-linked disorders. Now, before we go into there, some of these are called deletions, where chromosomes are missing, duplications, where there's an extra, inversions, where the chromosomes are turned around backwards, translocation, the one on page 307. You guys had to look this up, where you have a translocation of chromosomes 9 and 22, and we call this the y'all looked this up. It was on your last lab. Come on, people. Philadelphia chromosome. Good. So you have a translocation of part of the DNA from 9 to 22. And yeah, that causes some problems there. Causes cancer by activating a gene that leads to uncontrolled, cell progression. So those are all issues that can result from those abnormal chromosomal numbers. What else? Okay, before we go any further, let's talk about ploidy. So there's this term called aneuploidy, which means like an abnormal number of ploidy, meaning chromosomes. Okay, so we talked about some different examples. In plants, a lot of times, I don't know a lot of times, sometimes, I don't know the exact number, you see this effect called polyploidy. Polyploidy means extra ones. Now, if you're on 305 in your book on the top right, I'll kind of walk you through some examples of this. If you guys have ever extracted DNA from strawberries, y'all ever done that? Don't do that in 5th grade or 7th grade or something? We need to do that then. Remind me one day. Somebody bring some strawberries in. Strawberries are unique because they actually have 8 sets of DNA. So they are an 8N number. You know how we got 2 sets, one from mom, one from dad? Strawberries are 8N, which is very strange. Bananas are triploid or 3N. Wheat is hexaploid, 6N. So there's a lot of differences here. Now, I'll try to explain it without looking at my computer. Seedless watermelons. Why is this important? Who cares? Plants do it, we can't. They're a little different than us, right? Sometimes scientists think that it's beneficial in plants to help create certain levels of diversity. It seems like polyploidy, one extra or missing chromosome apparently disrupts genetic balance more than does an entire extra set of chromosomes in this polyploidy scheme. Now, I don't know that you see polyploidy anywhere in any animals. If y'all can come up with them and y'all send it to me, I'd like to know. But anyhow, how do we get seedless watermelons? How do you grow watermelons without seeds? They come from seeds. Who knows?
If you cross a 2 in with a 4.
You are a freaking genius. Yeah, how did you know that?
I was just saying a while ago.
Yeah, we talked about it.
God almighty, they listen to people in the other class. Yes. So you actually cross a watermelon. You cross a 4N watermelon with a 2N watermelon. I can't remember which one's male and female. Y'all have to look it up. I think the male's the 2N and the female's the 4N, if I'm not mistaken. But when you cross, so you have to plant some watermelon plants. So you're planting some watermelon plants here that are like a row of them that are like 2N, and you're planting some over here that are 4N. So they're going to cross-pollinate, right? And so their offspring are going to wind up being a mixture of that. They're going to be 3N. And 3N watermelons are like a mule. You know, you mix a horse and a donkey together, you get a mule. You got offspring, but they're not viable. They can't produce viable offspring. That's a simple analogy. So the same thing happens with the watermelons. You cross them, you get that 3N. They can't produce seeds. They can't produce viable offspring. And that's how we get our seedless watermelons, which is a beautiful thing because who doesn't like seedless watermelons, right? Well, people in here have no experience, but I don't like seeds in my watermelon. Any questions there? Now you know. I was actually listening to a podcast of a guy who works for a seed company, and he was going through the process and talking about which seeds he was mixing and matching. It's pretty cool. Syndromes. An organism possessing the identifying traits of a particular disorder. On the bottom of page 306, we have a karyotype. This karyotype is a karyotype of, if you look at it close, you can tell.
Which page?
306. Just by looking at the karyotype, you can tell that individual has
Down syndrome.
Down syndrome. We've got trisomy 21 here. You see how small those chromosomes are compared to the other ones at the top? So anyway, you're still talking about millions of base pairs, but the gene number is much less. So, Down syndrome, we know is trisomy 21. What are some other ones? In your book here on 307, girls, you can have 1X. It's called Turner syndrome. There are some physiological features that go with that. I'm not going to get into that. Girls, you can also have an extra X. Be careful not to search XXX on your computer or device, or something inappropriate may come up. Just ask the teacher down the hall from me who had to retire a couple years ago. It says it may occur, the XXX, once in approximately 1,000 live births, which seems pretty often. And it says no unusual physical features other than being slightly taller than average. So that doesn't sound like too bad a deal. They're still fertile. I mean, I don't even know if you know you had that. Maybe you just think, hey, I'm a little bit taller. Yeah. Turner syndrome is different. It's 1 in 2,500 births, and they're usually sterile, and there's some other features that go along with that. Guys, you can wind up getting 2 X's and a Y, which is called Klinefelter syndrome. Usually as the testes are abnormally small and the man is sterile, and some breast enlargement and other female body characteristics are common. Too many X's. All right, we need that one, but the other one, I don't know, it's too much. So these are things to consider, different types of chromosomal abnormalities. And the one last thing I want to talk about here is we have some other DNA that can cause problems too. Where's that DNA found? Good, in our mitochondria. Thank you for all participating. So we have DNA in our nucleus, and then we have DNA in our mitochondria. And there are different mitochondrial DNA disorders that you may want to be aware of, like mitochondrial myopathy, where the mitochondria lack the ability to make large quantities of ATP because they possess faulty DNA. Mitochondria do what?
They provide energy.
Yeah, they make ATP for energy. So if they're not making enough ATP and something in cell respiration, like the enzymes or proton pumps aren't working, then if you had mitochondrial myopathy, you would expect to have what symptoms?
Cells are dying. Tired.
No, cells aren't necessarily dying. They're just tired because you're not producing as much energy. I mean, it'd have to be really low for that. Now, I think I told you a while back about the baby who had 3 parents, right? No? Okay.
Yeah, you did.
Yeah, okay. So I want to say this was somewhere down in Mexico. If I'm not mistaken, they did this. I can't remember. But they took, so the mother had a mitochondrial disorder, which is problematic because the mom is going to pass on her mitochondria to the offspring. So they took someone else's egg, they extracted their 23 chromosomes, and then they put the DNA from the 23 from mom and the DNA of the 23 from dad into that egg. So they wound up with an egg that had mitochondrial DNA from one woman, okay? And they mixed the nuclear DNA from whoever the normal mom and dad was supposed to be. And then basically they had 3 different DNA contributors to this one offspring in order to prevent that kid from for sure having that mitochondrial disorder, because they're going to wind up with it from it being passed on. So win-win for the fertility specialists. You guys take care.